Welcome to City Steps. I'm your host, S.K. Rana, and joining me today on the steps of St. Paul's Chapel at Columbia University is MaryElena. Say hi.
Hi, guys.
Could you introduce yourself?
So, my name is MaryElena. I am a rising junior at Barnard College studying neuroscience and anthropology.
Neuroscience and anthropology.
Yeah, I actually came in knowing that I wanted to do something STEM-related, but I also really loved humanities and social sciences, and especially with it being a liberal arts college, I thought I would just give anthropology a go as a really eager first year in my first year fall, and I really just fell in love with anthropology and the way that it sort of teaches you to think differently about the world and from different angles. And then I took a medical anthropology class and found that it was sort of a complete opposite, but also the perfect complement to the STEM classes that I was taking that made me think about sort of the type of career that I might want to have in medicine and in STEM in the future as not just something that exists in an isolated bubble of the world of science, but also something that exists among humans, is practiced by humans, and just sort of gives it a whole new outlook on the world and why you do science as a person existing in a certain system at a certain time.
Wow, that's really cool. So I definitely want to get back to that, but let's get right off the starting block. What are you doing research-wise this summer?
Great question. So this summer I'm working at the New York State Psychiatric Institute in the Kellendonk lab, and that lab specifically focuses on schizophrenia. And there are different people in the lab that are focused, some more on like the cellular, some more on the behavioral element. And my project right now is a little bit of both. I'm new in this lab for the summer, so I'm still sort of getting my grounds on all the different protocols and sort of different bits and pieces. But right now I'm working primarily with this one mentor who's focusing on dopamine signaling in schizophrenia. So we're working primarily in the developmental side of the research with mouse pups and looking at how specifically dopamine 2 receptors operate in the different circuitry involved with schizophrenia in the mouse model.
Okay, that's interesting. So first, what does schizophrenia have to do with dopamine?
Based on current research, what we know is that schizophrenia can be caused by an overexpression of dopamine. And so that's why my lab and a lot of labs are focused on studying exactly how dopamine works. We know it's an overexpression, but we're trying to figure out exactly how that relates to different symptoms involving motivation. And if you could reduce that said overexpression of dopamine, then you could potentially have ways of addressing certain symptoms, whether they be cognitive, motivational, of schizophrenia in patients.
Wow. Okay, so when you say you work on mouse models, you're breeding mice?
Yes, so there are a few different bits and pieces to how the mouse model works overall. But we have multiple mouse lines, but they're bred specifically to be positive for certain genes that control the expression so that we can induce the same sort of D2, sort for dopamine 2 receptor expression that you find in patients with schizophrenia. So they have to be bred a certain way. And then after we breed them to exhibit a certain genotype, then when the mice are pups at this current point in the experiment, we actually inject a virus into the basal ganglia of their brains that will be used throughout the experiment to sort of track what's going on so we can better understand the circuitry of dopamine in the brain.
How does the breeding of the mice relate to dopamine?
Good question. So they're bred initially with a couple of different mouse lines. But basically what happens is when you inject the virus, that is what will induce this dopamine sort of expression that's supposed to model what happens in patients with schizophrenia. Like I said, right now a lot of the work is with immunohistochemistry to track sort of if the virus is hitting the right part of the brain. Basically, if we see the parts of the brain light up like a firework under the microscope, then we know it's doing what we think it is. And then once we know that the virus is hitting that indirect pathway, then we can modify the virus to be more of a treatment and know where to address the treatment areas.
So I'm still a little bit unsure. So does the virus just track how dopamine moves in the brain, or does the virus cause an overexpression of dopamine?
So the virus, we're using it to track because the virus itself has either the GFP or this protein called mCherry. So then when we shine a certain wavelength of light, whether it be UV light usually, then we can see different parts of the brain glow. So then we can track exactly where the virus is being expressed and if it's actually in the pathway that we're targeting. But the virus itself actually attacks the D2 receptors that are already in the brain. So it sort of does a little bit of both. It acts as a tracker, but with the way that the mice are bred, the virus reacts to cause the dopamine to the sort of transgenic. Like the mice are transgenic themselves, but then the virus...
What does transgenic mean?
Transgenic. So they're bred to have these certain genes that are not normally in mice. Mm-hmm. You use a certain technology called, I believe it's Krelok's technology, to actually insert the gene that you want that controls the dopamine expression. Okay. So, which is why actually just recently we had a little bit of a situation where we didn't see what we expected in the microscope, and one of the things that you have to go and check for is whether or not the mice are positive for this gene called Cre, because if it's Cre negative, then it can be used as a really good control to see how a normal mouse brain should look, but if they're Cre positive, then that's what sets off the rest of the chain of events.
Got it. So the mice already have this this possible trigger for overexpression of dopamine, and then the virus acts as like an initiator for this gene that's bred into them.
Yep, and there are other ways to do this sort of insertion besides the virus. From what I understand, before my time in the lab, my mentor also tried just a strictly breeding approach, but it can get pretty difficult with different genotypes and phenotypes and tracking breeding and controlling breeding, which is why we're now using a protocol that calls for injecting the mice at usually around prenatal day 0 or day 1 with this virus directly into their basal ganglia. And the tracking of dopamine right now is sort of, like I said, it's sort of the beginning phases of this longer set of experiment, and once you know through using the immunohistochemistry and the staining that you're hitting the right parts of the brain with the virus and that you can actually basically just make contact with it and manipulate it in some way, form, or fashion, then you can start focusing more on more like cognitive behavioral treatments and experiments down the road. But this is sort of where we are right now.
That's really cool. So here's a fun question. Did you have to get trained on essentially mice anatomy to do this, or mouse anatomy to do this?
Well, sort of, kind of. I went in not with a lot of experience in neuroscience specifically. Like I said earlier, I knew I wanted to do something in the STEM field when I came to college, but I sort of toyed around with the idea of like biology, maybe neuroscience, in the pre-medical tract. And I haven't had that much experience yet with neuroanatomy. So I spent lots of time over the past couple of weeks with an atlas of a mouse brain, sort of trying to figure out where things are and sort of what you're looking at, especially because things are they're just tiny, for lack of better words.
Yeah.
Especially when you're dealing with the mice. And it was actually my first day when I got to see what's called a perfusion, where you go and you drain all the after you sedate the mice. Then you drain all of their blood out, and then you push paraformaldehyde through their veins using the cardiovascular system that's still beating to basically preserve the brain. And then you take it out and you slice it. And my mentor was very patient to go through and point out what the tiny little dots and how you could tell this tiny little dot from another tiny little dot. But yes, you do generally have to be pretty familiar, especially when it comes to the microscopy. And so it took a lot of time, like going through and even now looking at the different landmarks. Especially in the initial like mouse surgeries, the first time I saw a pup surgery, I had no idea how I was going to learn how to hit a very tiny part of a very tiny brain. But through technology with coordinates and different anatomical markings, you figure it out.
So is this your first time doing research?
No, this is actually my second summer. I did research previously, still at the medical center uptown, in more of a biomedical engineering lab.
Is there a specific reason why you decided to do more of a neuro lab this year as opposed to last year where you did more engineering oriented?
Well, I took some classes specifically in psychology and neuroscience, just a little bit, sort of moving through the past academic year, and I declared a major in neuroscience as well. And I absolutely loved my first lab. It was in infection control, and I got to learn a lot about sort of microorganisms and how it works. And a lot of what I learned in my first lab was just sort of how to be comfortable working in a lab, whether it's developing technical skills or developing just the confidence to work on your own and sort of supervise what you're doing. And I absolutely love the environment, and I wanted to find a way to incorporate sort of the things that I was learning in my classes, moving more into the neuroscience field. more so in a heavy way than I had before.
So you mentioned earlier that you are a neuroscience major and an anthropology major, so you're a double major.
I'm an anthropology minor, actually.
You're an anthropology minor.
Yes.
Okay, so you're a neuroscience major, anthropology minor. How do you see your major and minor influencing what you want to do in the future?
Great question. So I, as of right now, know that I want to do something in the medical field, and I, as of right now, would actually like to do something that sort of incorporates the two in some form or fashion, whether it be in the form of some sort of combination of degrees in both. Something that would allow me to practice medicine as well as practice anthropology. But one thing I've looked into seriously is doing some sort of MD, PhD program, either conjointly or separately, and doing something that would allow me to practice medicine, but also as an anthropologist. And I would do my research in specifically medical anthropology, and sort of studying how science and technology influences things, like the culture and society that we live in, and vice versa, because those things are never entirely separable. So I think that's given me, if nothing else, just a new perspective with which I hope to practice medicine in some capacity. But I also am exploring the idea of practicing medicine as an anthropologist and vice versa.
So what is medical anthropology?
So medical anthropology, sort of boiled down, is looking at medicine through a non-STEM type lens. So you're not necessarily looking at just, and not to say that all medicine is like this, I might be simplifying it a bit, but not that you're just looking at medicine as, treating cells or treating a system, but actually treating people who are embedded in these different sociocultural systems and come from different economic backgrounds, like how different types of treatments might not necessarily be implemented the same way in different cultures, different regions of the world, and sort of investigating also how medical professionals can be aware of this, that although they may be licensed to practice in XYZ country, that doesn't necessarily mean that you are prepared as a medical practitioner. There's still always this element of like learning and adapting culturally and socially.
Because every culture has different social norms, and medical practices can often are inherently invasive, and you need to adapt those to different social cultures and social frameworks.
Oh yeah, exactly.
Wow, that's really cool. So you're considering an MD, PhD, or something of the sort?
Yeah, something like that. I know there are lots of different programs that sort of incorporate, even if it's not in media anthropology, sort of more the social science and humanities side, whether it be a program in narrative medicine in conjunction or around the same time as an MD and programs like that, definitely.
That's so cool. Has doing research the past 2 summers influenced your decision to pursue a PhD, possibly?
Yes, I believe so, specifically in terms of this lab that I'm in right now. My mentor is actually an MD, PhD. So he's a practicing psychiatrist and also a neuroscientist. And although his research in neuroscience might not necessarily be the type of PhD that I might pursue, whether it be neuroscience or anthropology, it's been great to have someone who's gone through the process of getting an MD and a PhD in something.
Which is really hard. That's 7 to 9 really long years.
Oh yeah, and just talking to him about his experiences, and it can honestly be a little reassuring too, just to hear him talk about like going home to see his family and his daughters, to know that I think sometimes it's a little easy to get wrapped up in the stress or the classes or the applications, and not think about the fact that it is possible to have sort of a life, not only a life outside of science, but also a life that can sort of be complemented and balanced with science. And so him talking about just all the other things that he does and is passionate about is, I think, inspiring, and just a good lesson that, science is great, PhDs, MDs, they're all great, but there are also other things in the world, and it's not necessarily always a question of having to choose between them.
Wow, that's a really great sentiment and a really great stopping point and wrapping up point. Thank you for letting me interview you, MaryElena.
Of course, anytime.
And this is our 4th episode of City Steps. Thank you for joining us.